Chapter 16. Repetitive Sequences, Gene Duplication, and the Varieties of Genomic Variation. Man's creative genius flourished only when his mind, freed from the worry of daily toils, was permitted to entertain apparently useless thoughts. In the same manner, one might say with regard to evolution that natural selection merely modified, while redundancy created. Big leaps in evolution required the creation of new gene loci with previously non-existent functions. Susumu Ono. The Big Picture. Several types of changes in DNA sequences, including production of repetitive sequences, unequal crossing over, gene duplication, point mutations, and more, act on the genome in a synergistic manner. Together, they have extraordinary organizing power. The UES recognizes that genomes have incorporated several general tools that facilitate, leverage, and accelerate genome evolution. Darwin's original concept of accumulation of infinitesimal changes must be supplemented with these widespread and now well-known mechanisms of evolutionary change. The modern synthesis focus on point mutations as the major source of new alleles fails to recognize numerous additional mechanisms of genomic sequence evolution. In many cases, several mechanisms have combined effects on the genome that are far more significant than any one mechanism alone. Both genotypes and phenotypes can change in a variety of ways that leverage previous innovations. This can result in rapid addition of structures and functions that did not need to arise incrementally, but are often subsequently refined in an incremental fashion. Sometimes during DNA replication, the growing strands can mispair with the template strands. This mechanism, known as slipped strand mispairing or replication slippage, represents a fundamental way that DNA sequences can expand into simple repetitive sequences that are subsequently diversified by other mechanisms, such as point mutations. Gene duplication represents one of the most widespread and significant types of genomic evolution. Duplicated genes played a crucial role in the evolution of diverse forms of life, ranging from prokaryotes to plants and animals. Most of the genes in animals are members of multi-gene families that arose by gene duplication events. Duplicated genes lay the groundwork for new innovations that have far more organizing power. The keen sense of smell exhibited by dogs is largely a consequence of extensive gene duplication events in olfactory receptors. The mammalian immune system provides a quintessential example of the sheer organizing power of the combined effects of gene duplication, VDJ recombination, and somatic hypermutation to generate extraordinary diversity.